change the brightness. So, good morning, everyone.
Tea or coffee? Tea or coffee?
Anything small. Increase it. So, good morning, good morning. Am I audible? Sound is coming. Hello. Voice not audible?
Yes.
Voice is not coming, students are saying.
Yes.
Audible? Voice is coming now? No voice. And increase the brightness also. Brightness is very dull. My mic is turned off, not turned on, no? It is turned on, no?
No, sir.
Check from there. Hello, hello, hello. Is coming? Once again. It's all right? Coming? Voice is coming? Not coming? Not coming. YouTube channel coming, but Zoom not coming. Check in Zoom. There it is going. So check from here. Either it is muted from here.
Yes.
So unmute it.
Yes.
I hope now it is all right. Now okay. Brightness. So we are starting with lipid. Okay. So now give your attention. See the things. So lipid is classified into 3 different categories. One is simple lipid. Another one is conjugated lipid. And then another one is derived lipid. So 3 types of lipids are there. Simple, conjugated and derived. So if you talk about simple lipid, it is of 2 types. One is called true lip fat, or it is also called neutral lipid. And another one is waxage. Right? So if you talk about simple lipid, it is of 2 types, true lipid or true fat or neutral lipid and what? Waxage. Okay, ji, it's all right? Okay. Now, if you talk about this true fat or neutral lipid, what happened? This true fat or neutral lipid is made up of 2 components. One component is glycerol and another component is fatty acid. So if someone asks you true lipid or neutral fat or true fat, made up of what? Made up of 2 components, glycerol and fatty acid. Clear? So if you talk about glycerol, we also call glycerol as what? Glycerol is what? Trihydroxypropane. Trihydroxypropane. This is alcohol. Glycerol is a what basically? Alcohol. Not we just started. So nothing left. Getting the idea? So one student asking, I just joined, anything missed? Nothing missed, we just started it. So trihydroxypropane is the another name of which one? Glycerol, and here it is what basically? Fatty acid. So this glycerol or trihydroxypropane is what? Alcohol. Getting the things? It is an example of alcohol. Now what is about the waxage? How wax are different from the true fat? Just give your attention. If we talk about wax, it is also made up of two components. One is alcohol, other than glycerol. Plus fatty acid. So if someone asks you a question, how wax are different from true fat? What the answer will you give? Okay, in true fat, which alcohol is there? Glycerol is there, plus fatty acid is there. But in waxage, who is not there? Glycerol is not there. Some another alcohol is there in place of glycerol. Getting the idea or not? And who is there? Fatty acid is there. So how true fat and waxage differ from each other, you got the idea or not? Clear in the mind? So say both wax and true fat are example of which lipid? Simple lipid. Done? Clear? Now I am telling you about individually about glycerol, individually about fatty acid. These are also called a part of simple lipid. Simple lipid, glycerol, fatty acid. So we are going to talk about glycerol now. So have a look. Glycerol. So if we talk about glycerol, the structure of glycerol is what basically? It is trihydroxypropane. This is the structure of the glycerol. And what is this basically? Trihydroxypropane. Glycerol, or it is what basically? Or trihydroxypropane. And now I am going to tell you about fatty acid. So this is about the glycerol, and then we are going to talk about which one? Fatty acid. The another component of simple lipid. Another component of true fat. Fatty acid. If you talk about fatty acid, we can say it as what? RCOOH. RCOOH. Where R is what basically? Alkyl group. R is what basically? Alkyl group. Getting the idea or not? R is alkyl group. Done. Now if we tell about R is equal to, you can say methyl. R is what basically? C2H5. You can say R equal to what? CH3, CH2, CH2 dot 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 COOH. Getting the things or not? Simply, these are R basically. So if we'll talk about the number of CH2 in R equal to what basically? In R, if you talk about CH3, and CH2 number is what basically? CH2 number is 1 to 19 carbon. 1 to 19. Able to understand or not? So it methyl, ethyl, propyl, butyl, getting the things or not? So number of CH2 is how many in R group? NCERT wrote 1 to 19 CH2 group is there. So I'm just writing some example of fatty acid. Example, I'm writing palmitic acid. So if you talk about palmitic acid, how many carbon is there? 16 carbon, including carboxyl carbon. Arachidonic acid. It is how many carbon fatty acid? It is 20 carbon fatty acid. 20 carbon is there. And these are including which one? COH. These are including COOH. It is 16 carbon and it is how much carbon? 20 carbon. Getting the idea, understanding the idea. So these are the what basically fatty acid. So R is what basically a long chain hydrocarbon. And who is joined here? COH is joined. It is a fatty acid. R is what? It is a long chain hydrocarbon, alkyl group, and along with that, who is attached? Carboxylic acid is going to join. And that is the reason for fatty acid, what I have written here, RCOOH. Are you getting the idea or not? This is the fatty acid. Clear? Done? Now we are going to talk about type of fatty acid. So the next slide I am just changing. I am going to talk about type of fatty acid. So if you talk about the fatty acid, it is of two types. One is essential fatty acid and another one is non-essential fatty acid. So if you talk about essential fatty acid, such kind of fatty acid who not synthesize in our body, who not synthesize in our body are called what basically? Essential fatty acid. So not synthesize in body and have dietary requirement. That fatty acid require in which one? Your diet. Have dietary requirement. Got the idea? If it is not in your diet, you will got the deficiency of this particular fatty acid. And that is the reason. And non-essential fatty acid what? It synthesizes in our body. So out of all available fatty acid, only 3 are essential. I am writing the name of essential. Example of essential. The first one is what? Linoleic acid. The second one is linolenic acid. And the third one is arachidonic acid. So these are basically the essential fatty acid. Who are the essential fatty acid? Who are the essential fatty acid? Linoleic, linolenic, and arachidonic. So these are the essential fatty acid. They require in our diet. They not synthesize in our body. And many times when you got certain problems in your body, doctors give some essential fatty acid. Many times doctor prescribe you to eat which one? Omega-6. Heard about the name? Omega-3. So these are what basically? Essential fatty acid. Getting the idea or not? Omega-6, omega-3, these are what basically? Essential fatty acid. So sometimes when our food not have that particular fatty acid, we just got the deficiency. So for fulfilling, getting the things or not, right? Okay, ji, done. So linoleic acid, linolenic acid, arachidonic acid, these are what basically? These are what basically? Essential fatty acid. They need in our food. They have dietary requirement. They not synthesize in our body. And other than these 3 fatty acid, whatever the another fatty acid is there, our body made it. They are synthesized in our body, and that is the reason it is non-essential in nature. Got the idea? Clear in the mind? Now we are going to deal the further classification of fatty acid. If we talk about fatty acid, we can further classify it into 2 categories. So one is called saturated fatty acid and another one is unsaturated fatty acid. Unsaturated fatty acid. If you talk about the saturated fatty acid, no double bond is present. present in which one? R group of fatty acid. Double bond is not present. Got the idea? In unsaturated fatty acid, what happened? One or more double bond is present. In R group of the fatty acid. Got the idea? First difference is this between saturated and unsaturated fatty acid. If you talk about saturated fatty acid, it have high melting point. High melting point. Unsaturated fatty acid have low melting point. Low melting point is present in which one? Unsaturated fatty acid. If you talk about example of saturated fatty acid is what basically? Ghee. And example of unsaturated fatty acid is oil. Gingerly oil. It is written in your NCERT, na? It is unsaturated fatty acid. It is liquid in nature. It is liquid in nature. Ghee is going for solidification, na? So who gets solidified are saturated fatty acid. And who are present in liquid form is which one? Unsaturated fatty acid. Because unsaturated fatty acid have low melting point. And saturated fatty acid have high melting point. It will melt, but you have to increase the temperature for it. It will melt in summer season. Getting the things or not? But remain in solid in which one? Winter season. It remain liquid in both summer and which season? Winter season. So you can easily differentiate between which one? Saturated and unsaturated fatty acid. So most of the animal source fatty acid are what? Saturated. Most fatty acid from animal source are saturated. And most fatty acid from plant source are unsaturated. And if we talk about unsaturated fatty acid, it is of two types. Unsaturated fatty acid is of two types. One is MUFA, another is PUFA. MUFA and PUFA. MUFA is what? Monounsaturated fatty acid. PUFA is what? Polyunsaturated fatty acid. So number of double bond is more in PUFA. Able to understand? And who is the best fatty acid for our consumption? PUFA. As many double bond is there, it is better for health. So who is better for consumption? PUFA, polyunsaturated fatty acid. Saturated fatty acid is not good for health. Got the idea? Because saturated fatty acid easily converted into cholesterol. Easily converted into cholesterol. So it is not good for health. So earlier, you will, I think many of you are aware about a product came of saturated fatty acid in market. Product name was Dalda. Actually, that is vegetable ghee. Are you aware about the name of Dalda? Getting the things? That is what basically saturated fatty acid. Nowadays, you are finding that is very less in consumption. Usually, people don't use it. Avoid it. Because it is saturated fatty acid. It produced by hydrogenation of vegetable oils. Is it clear? So when we go for hydrogenation in unsaturated fatty acid, who will form? Saturated fatty acid will produce. Got the idea? Clear? And saturated fatty acid easily convert into which one? Cholesterol. And you know, cholesterol level when increase, it is not good for our health. So that is the reason it is recommended what? We have to go for which one? PUFA, like oleic acid. Getting the things or not? So you will talk about what happened in like olive oils. Some of the fatty acid are what basically? It is polyunsaturated fatty acid. Now we are moving ahead. So fatty acid over. Now glycerol also over. Now I am telling how glycerol and fatty acid join together. Look here. It is glycerol, trihydroxypropane. And here I am just writing about fatty acid. Here I am writing R1. I am writing here R1COOH. So see how they are joining together. Fatty acid join with glycerol. By which bond? Ester bond. Esterification occur. And who is releasing here? Water is releasing. When one ester bond form, one water molecule release or not? So they join by which bond? Ester bond. Esterification occur. So when one fatty acid join with a glycerol, who is producing? Monoglyceride is producing. Who is producing? Monoglyceride is producing, and water is removing, means dehydration reaction occur. And the bond produced between fatty acid and glycerol is what? Basically ester bond. Getting the things or not? Ester bond production occur. So one ester bond formation occur, and this structure is called monoglyceride. Who is forming here? Monoglyceride. When two fatty acids join, R2COOH, here also a water will remove 2H2O. Now it is called diglyceride. Now it is called diglyceride. Now if 3 is producing R3COOH, 3 fatty acid is joining. Getting the idea? It is forming triglyceride. And how many water molecule remove? 3 water molecule remove. So this is the structure at present on the board is a which one? Triglyceride. So in triglyceride, how many glycerol is involving? 1 glycerol. How many fatty acid involved? 3 fatty acid. How many ester bond? 3 ester bond. And how many water will remove? 3 water molecule. And this structure which is formed here is what? Basically triglyceride. So either it is a monoglyceride, diglyceride or triglyceride. They are what? Basically true fat. They are true fat. I told about the definition of true fat. In true fat, who is present? Glycerol and fatty acid are present. Getting the idea? So these either monoglyceride, diglyceride or triglyceride, these are the example of which one? True fat or neutral lipid. Clear in the mind? It is over. Right? Now we are moving about the waxage. Usually people use every day wax, 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 but they don't know what is wax. Wax is a which kind of lipid? Simple lipid. First thing. How wax is forming? When fatty acid join with alcohol other than glycerol. How wax is forming? When fatty acid join with which one? Alcohol other than glycerol. Then formation of wax occur. So some of the example of wax I am writing. V wax. You have seen the honeybee comb. So, right? So there who is present? It is made up of which one? V wax. Got the idea? Now this wax is also present in your ear. In ear wax is there or not? Which gland secretes wax in your ear? Write the name. Which gland? Not sebaceous. It is which one? Curiminous gland, na? I forget zoology, but not that much. You don't manage to know about the name of gland. It is which one? In your ear, who is there? Curiminous gland is there, na? Who are producing which one? Wax. And you touch your face. You find here who is there? Smooth structure is there. Smooth you are feeling, na? These are also wax. These are the release product of which one? Sebaceous gland. These are what? Sebaceous gland. Getting the idea, understanding the idea, getting the thing. So V wax, sebum, curiminous gland present in your, getting the thing, ear, these all are producing wax. Wax are also present in which one? Surface of leaf. So these all are simple lipid. Getting the things or not? Ear wax, you can say sebaceous gland secretion, these are the wax. So these are what basically? Simple lipid. How they are forming? They are forming when fatty acids joined by which one? Alcohol other than glycerol. Got the idea? I will send you the QR code. So lastly, every slide will go in your mobile phone. Getting the things or not? If you scan it. So that is the reason I am not giving you much time to write. Getting the things. Lastly, I will send you the QR code. Now, who is over? Simple lipid is over. Now we are going to talk about conjugated lipid. In conjugated lipid, what happened? Along with lipid component, non-lipid part also present. Lipid plus non-lipid component together forming conjugated lipid. I am just writing example. Phospholipid. Phospholipid is an example of conjugated lipid. I am writing glycolipid. Glycolipid is a conjugated lipid. I am writing lipoprotein. Lipoprotein is an example of conjugated lipid. In phospholipid, who is there? Along with lipid, who is there? Phosphate is there or phosphoric acid is there. So phosphoric acid is what? Non-lipid part. Glycolipid, along with lipid, who is there? Carbohydrate is there. Carbohydrate is non-lipid part. So it is glycolipid. If you talk about lipoprotein, along with lipid, who is there? Protein is there. So when along with lipid component, non-lipid component is also present, then such kind of lipid is called what? Conjugated lipid. And you are well aware about these phospholipid. These are the phospholipid present in your plasma membrane. Present in plasma membrane. Got the idea? So this phospholipid, I already told in the plasma membrane, कि phospholipid have two polarity. What? Head is hydrophilic and tail is hydrophobic. Right, and that is the reason it have both positive and negative, water loving affinity and water hating affinity. And that is the reason we told it what? Amphipathic. Is it clear in the mind? If I draw the structure of phospholipid, how to draw it? I am drawing their structure. Look it here, how the phospholipid is forming. So here it is glycerol. Here it is glycerol. And when this glycerol join with which one? Fatty acid. Look here. Here R1 COOH. Ester bond. R2 COOH. Ester bond. So with glycerol, how many fatty acid chain join? 2 fatty acid chain join. Now in place of third fatty acid, who is joining here? H3PO4. Who is joining here? H3PO4. Means which one is joining here? Phosphoric acid. Who is joining here? Phosphoric acid is joining. So when glycerol join with how much? 2 fatty acid and 1 phosphoric acid. Who is forming here? These phospholipid is forming. Getting the idea? Getting the idea? So how the phospholipid forming in their structure, you got the things? This is the glycerol. With this glycerol, 1 chain of fatty acid, 2 chain of fatty acid. It is R1, it is R2. And in place of another chain of fatty acid, who is joining? Phosphoric acid is joining. And then it is going to produce which lipid? Phospholipid. So how the phospholipid is producing, you got the idea, understand the idea or not? And now NCERT wrote about an example of phospholipid. What example written in NCERT? Lecithin. Lecithin is an example of which kind of lipid? Phospholipid. How to write the structure of lecithin? Look here. Look here. Here it is phosphoric acid. After that, with this phosphoric acid, who is joining? CH2, CH2, N, CH3, CH3, CH3. So this is also a phospholipid, and the name of this phospholipid is which one? Lecithin. Lecithin. Examiner will ask, what is lecithin? It is a phospholipid. It is a phospholipid present in membrane. You got the idea? So this is the structure of a lecithin. Done? Over. Now we are going to deal about conjugated lipid. Phospholipid over. And conjugated lipid over. Now we are going to deal about which one? Derived lipid. Now we are going to deal about which lipid? Derived lipid. If we talk about the derived lipid, it is of two types. Esterol, and another type of derived lipid is which one? Terpenes. Terpenes. So esterol obtained from different sources. Example of esterol is what? Phytosterol. It obtained from which one? Plant. Ergosterol. Sources of ergosterol is which one? Fungi. Zoosterol. Sources of zoosterol is animal. I am writing here sources. An example of zoosterol is what? Cholesterol. Excuse me. So example is what basically? Cholesterol. Cholesterol is an example of which one? Zoosterol. Another example of sterol in nature is what? Sex hormones. Is a steroidal, na? It is a steroidal. So who are steroidal in nature is what basically? Sterol. Getting the things or not? Getting the idea? Clear? An example you can say estrogen, progesterone, testosterone, even esterol produced by which hormone? Adrenal gland. Corticosterol. You studied, na? Corticosteroid. Adrenal cortex. Is it? So these are the some of the example of which one? Esterol. This cholesterol is present in plasma membrane also, provide stability to plasma membrane. And now finally I am going to talk about terpenes. Example of terpenes is what? Monoterpenes, diterpenes, etc. etc. So who is over? Fat is over. Lipid is over. Done? So just I have given an idea about simple lipid, derived lipid, conjugated lipid, or simple, conjugated and derived. Simple lipid is true fat and waxes. True fat is made up of glycerol and fatty acid. Waxes are made up of fatty acid and alcohol other than glycerol. Conjugated lipid have what? Lipid plus non-lipid component. And derived lipid have two types. One is what? Sterol, another is terpenes. Sterols, if obtained from plant source, phytosterol. From animal sources, it is zoosterol. From fungal sources, it is ergosterol. And lecithin is an example of which one? Phospholipid. Lecithin is an example of which one? Phospholipid. Got the idea? Done? It is a derived lipid. So lipid is over. After lipid, we are going to discuss about metabolites. We are going to deal about metabolites. What is these metabolites? So these are intermediates of biochemical reaction. So intermediate of biochemical reaction is called what? Basically metabolites. In a reaction which occur in your body, two type of reaction occur. One is anabolism, another is catabolism. So during anabolic reaction and catabolic reaction, different intermediate who produce are called what? Basically metabolite. Even you can say metabolite to the end products to a intermediate in which one? Metabolic reaction. Right. So if we talk about these metabolite, it is of two type. One is primary metabolite and another one is secondary metabolite. So if we talk about primary metabolites, where it is present? So primary metabolite present in all organism. No exception is there where primary metabolite is not present. In every living organism who is present, primary metabolites are present. Either it is bacteria or it is fungi or it is plant or it is animal, everywhere it is present. Done? So you can say in animal, what kind of biomolecules you can find during a chemical reactions. These all are example of which one? Metabolites. Now secondary metabolite present in plant. It also present in fungi. It also present in microbes. But these secondary metabolite absent in animal. In animal, secondary metabolites are not there. In both plant and animal, primary metabolites are there, but secondary metabolites are absent in which one? Animal. Now, what are the some another differences? So if you talk about primary metabolite, have known physiological function. Known physiological function. Known physiological function. What function a primary metabolite do, we know about it. Here, mostly unknown. Unknown physiological function. Means we have no much idea about why secondary metabolite are produced in plant and bacteria or fungi. We don't have much idea about their physiological role in their body. So they have some ecological role and they also used in human welfare. So we use the secondary metabolite for usefulness for a human being. But secondary metabolite, why it is producing in the plant? We have very little idea about it. Some ecological role is there, that secondary metabolite, and that ecological role is what? Some secondary metabolite are very bitter in taste, and when any animal eating that plant part, either the plant animal get irritated by that particular bitter taste, or definitely some physiological problems going to arise, so it avoid it to not eat. An example in ecology you have studied cardiac glycosides. These are what basically a secondary metabolite, and it helping the plant to protect. So these are the some ecological role. But what are the physiological role of the most of the secondary metabolite? We don't know about it. Getting the idea? Now I am telling about the example of primary metabolites. Example of primary metabolites I am writing. Example is amino acid. Example is fatty acid. Example is glycerol. Example is glucose. Example is ribose. Example is nitrogenous base. That is adenine, thymine, cytosine, guanine, uracil. Another example is what? Cholesterol. Means these example you can write, board will fill. Got the idea? Means all the chemical compound, biomolecules present in your body comes under which metabolites? Primary metabolites. In your body, amino acid present or not present? Yes. Fatty acid is present or not? Yes. Glycerol present or not? Yes. Adenine, thymine, cytosine, guanine, uracil present or not? Yes. DNA, RNA is there, so it is present. Got the idea? Cholesterol present or not? Yes. So whoever present in your body are example of which metabolite? Primary metabolite. You got the idea? Understand the idea? So will you find any confusion to identify who is the primary metabolites? Yes. Tell me. Will you confuse? Any confusion occur to identify who is primary metabolite? No confusion. Now I am telling you about what? Secondary metabolite. And from here question is going to ask in exam. Secondary metabolite. It is very important for examination point of view. So first of all I am writing some example of secondary metabolite, category of example pigments. Alkaloid, toxin, drugs, lactins, terpenes. After that you can say polymeric substances. So these are some of the list of secondary metabolite present in all organism other than plant. It's not necessary in each organism every secondary metabolites are present. Getting the things or not? It is not necessary. But secondary metabolites, pigments, example what? Carotenoid is a secondary metabolites present in plant. Anthocyanin is a pigment present as a secondary metabolites. Alkaloid, what are the name of alkaloid? Morphine. You can say codeine. So these are secondary metabolite. Now question arises, why morphine is present in plant? What is the role of morphine in plant? We don't know about it. But morphine have used in role in our welfare. Na? How morphine is helping us to make painkillers? Painkiller, getting the things or not? So morphine, codeine, these are what basically? Codeine is present in cough syrup. It is present in which one? Cough syrup. And it used in which one? When you consume cough syrup, it creates a drowsiness, na? Drowsiness, you just sleep. Got the idea? So these are what basically some of the what basically secondary metabolite function. Toxin. What are the examples in the toxin? Ricin. Another example of toxin is what? NCRT example, do it which one? By heart. ARUN. These are what basically secondary metabolite. Got the idea? Not ARUN, it is ARUN. Next is what? Drugs. Who is in the drugs category of secondary metabolite? Drug. Tell me. Curcumin. Another who comes in the drug category is vinblastine. Right? Vinblastine. In lactanes, who is coming? Concolvin A. It is what? Lactanes. Terpenes, monoterpene. Diterpene. Another list is polymeric substances. Who is there? Gums, rubber. These all have a role in human welfare. Cellulose. And another category I am writing, essential oil. Essential oil. Who is coming in this category? Lemongrass oil. Lemongrass oil. So these all are the list of which one? Secondary metabolite. You do it by heart. Like 2 2s are 4. Getting the things. From here, questions is going to ask in your NEET exam. How the examiner will ask the question? They will ask a question. Vinblastine is a toxin, drug, alkaloid, or lactanes. So you have to choose any one. So vinblastine, if you don't mug up, it is a drug, answer become wrong. So here simply mugging up activity require which secondary metabolite and what are the particular example of that particular secondary metabolite. These are not present in animals. And so what I am telling? I am just telling another secondary metabolite example. An example, caffeine. It is not written here. Caffeine is also a which one? Alkaloid. Caffeine present in tea and coffee. So it have a role in human welfare, but why plant is producing? So for every secondary metabolite, we don't have much idea about it. What are their physiological role in plant? Why plant is producing? What is the physiological role of caffeine in plant? We don't have much idea. Getting the idea? Is it clear in the mind? But it are used in the which one? Welfare of the human beings. So you got the idea about which one? These secondary metabolites. Is it clear in the mind? Over. Now we are moving ahead. Now we are going to deal about biomolecules categorization. So biomolecules we categorize into two categories. One is biomicromolecule. And another category is which one? Biomacromolecule. Biomicromolecule and biomacromolecule. How to differentiate between the two? First, based on their molecular weight. So biomicromolecule have a molecular weight in the range of 18 to 800 Dalton. If you talk about the molecular weight of biomacromolecule, it is more than 1000 Dalton. So who have a molecular weight more than 1000 Daltons will comes under biomacromolecule, and who has a molecular weight in the range of 18 to 800 Daltons will come under what? Biomicromolecule. So except lipid, all biomicromolecule is present in acid soluble fraction or acid soluble pool. Getting the things or not? If you talk about bio-macromolecule, they are present in acid insoluble pool. Insoluble pool. Because they have a bigger size. So all bio-macromolecule is polymeric substances. Polymeric substances means they are a polymers. Examples I am writing of biomacromolecules are protein, polysaccharide, and another example is which one? Nucleic acid. So these all are what basically? Polymer. Polymer, na? Protein is a polymer of amino acid. Many amino acids join and form a chain called protein. Polysaccharide, monomeric unit is glucose. Many glucose join together and what? Forming a polysaccharide. Nucleic acid, what is the monomeric unit? Nucleotide. Many nucleotides join together and form what? Nucleic acid. And that is the reason they all have a very high molecular weight. So the molecular weight of protein, polysaccharide, because it is a polymeric substance, chain of the monomeric unit. So due to that reason, either protein, polysaccharide, nucleic acid, they have molecular weight greater than 10,000 Dalton. Much more than the required level for definition. So protein, polysaccharide or nucleic acid have a molecular weight greater than 10,000 Dalton. Definition is saying if it is more than 1,000, it is biomacromolecule. These have more than 10,000. So very high molecular weight. What is the reason behind this high molecular weight? Because they are what? Polymers. Many repeating units join together, and it become a long chain, and due to that reason it have a high molecular weight. Is it clear in the mind? Done? Over? Right? Now I am writing one line, that is lipid have molecular weight less than 800 Dalton. So according to molecular weight, lipid comes under which one? Biomicromolecule or biomacromolecule? Biomicromolecule. According to molecular weight, it comes under which one? Biomicromolecule. So you can say lipid is only biomicromolecule present in acid insoluble pool. And who present in acid insoluble pool is called what? Macromolecule. Exception is there, that is what? Lipid. And that is the reason NCERT wrote, lipid not strictly a which one? Biomacromolecule. Because it is present in acid insoluble fraction along with biomacromolecule. But its molecular weight not justify the definition of the macromolecule. Now question arises, if lipid is small, beside of that, why it is present in acid insoluble fraction, who is large like polymers, bigger size? I can understand that it is present, it is not passing through the filter. But lipid is small in size. Beside of that, why lipid is present in acid insoluble fraction? So one student have given a very good answer. So you see, lipid is present in plasma membrane. It have both hydrophobic and hydrophilic end. And when we break the plasma membrane, lipid is forming the vesicle. And these vesicle don't pass through the filter. And that is the reason lipid is present in which fraction? Acid insoluble fraction. Beside of that, it have a small size. So what the answer will you give? Ki lipid forming a vesicles. And these vesicle can't pass through the filter. And that is the reason, beside of that, it have a what? a small size. Getting the things? It is present in which one? Acid insoluble pool. Beside of a small size, it is present because it is present in the form of which one? Vesicles. So will you keep this line in mind or not? So if anybody going to ask you why lipid is present in acid insoluble fraction, are you in position to answer the question? Because the lipid who are present in membrane have two polarity, hydrophobic and hydrophilic. And when we break the membrane, they not remain as a chain, they are forming a ring. They are going to form a vesicle like this. And these vesicles, how it pass through which one? How it pass through which one? Sieves or pores. And that is the reason it is present in which one? Acid insoluble fraction. Getting the idea? Is it over? So you got who are coming under biomicromolecule and macromolecule. So except these three, protein, polysaccharide and nucleic acid, I am asking, glucose is biomicromolecule or macromolecule? Glucose, micro or macro? Micro. Amino acid, micro or macro? Micro. Only macro is what? Protein, polysaccharide and nucleic acid. Done? Over? Who are polymeric forms. Now we are starting our story with which one? Bio, macro, molecule. And first thing we are going to discuss in this category is which one? Protein. So we are going to discuss about protein. Give your attention. Protein. So first of all, you keep in mind, it is a heteropolymer. Protein is a heteropolymer. Why heteropolymer? Because more than one monomeric unit are there. Getting the things? More than one monomeric unit are present in protein. Na? So it is made up of 20 different amino acids. So in a protein, how many different amino acids are participating? 20 different amino acids are participating. So monomeric unit is 20 type of amino acid. And that is the reason we are saying what? Heteropolymer. If it is only one type of amino acid, many amino acids join and form a protein, we call it what? Homo. But here participating amino acid is how much? 20 different amino acids. And that is the reason we are saying protein as a which one? Heteropolymer. You got the idea? It is a polymeric substance, but monomeric unit is not of single type. 20 possible type of monomeric unit is involving in the protein formation. So based on amino acid, if you talk about amino acid, NCERT wrote it, there are two type of amino acid are there who participate in protein synthesis. One is which amino acid? Essential amino acid. Another is what? Non-essential amino acid. You know the definition of essential well. Who need in diet? Who require in diet? Means not synthesized in body. In body is called essential amino acid. And non-essential amino acid who synthesize in body? Is called non-essential amino acid. So out of 20 amino acid, 10 is essential and 10 is non-essential. And what is the source? What is the source? Source is your chemistry book, biochemistry. Got the idea? In your biochemistry book, it is written, a table is given in that chapter. Amino acid who are star marked are essential, who is non-star marked is non-essential. So who is essential, who is non-essential? In that particular chapter of biochemistry, chemistry sir will teach it. I am not going to tell you the list. because it is not mentioned in your bio books. It is mentioned in your chemistry book. So sir will take care there. I am just simply telling out of 20, 10 are essential, 10 are non-essential. And why I told? Because many people who teach, sometimes someone saying 9 are essential, 8 are essential, semi-essential. So these all nonsense not going to ask in what exam. What is NCERT centric data is there, that is going to ask in exam. Getting the idea? Is it clear? It is all right? So essential and non-essential definition, you got the idea? Yes. Now I am writing the function of protein. What function performed by protein in our body? Function of protein. So protein is forming which one? Intercellular ground substances. Ground substances or matrix. Protein may act as enzyme. Protein may act as hormone. Protein may help to fight against the diseases. Got the idea? If you talk about the another function of the protein, protein may act as a receptor. Protein may have a role in which one? Transport. So see, these are the variety of role in our body performed by which one? Protein. So tell me the example. Intercellular ground substances formed by which protein? Collagen. Collagen is a protein who are forming which one? Intercellular ground substances. Almost all enzymes present in your body are made up of protein. Exception is there, ribozyme. So NCERT wrote a data here, what name here? Trypsin. Trypsin is an enzyme. Hormone, many hormone are proteinaceous. Example, insulin. Antibody is fight against infection. Antigen or infection. If you talk about protein, also used as a which one? A receptor. Getting the things or not? A receptor. Is it clear? So if we talk about the receptor, you can say insulin receptor. Insulin receptor is made up of which one? Protein. With which who bind? Insulin bind. Able to understand? Transport, protein have a role in transport. Very good example is GLUT4. Glucose transporter 4 protein. It have a role in transport of the glucose. Getting the thing? So protein is serving variety of role in our body. Getting the things? Have a role in ground substances, acting as enzyme, acting as hormone, antibody, receptor, and have a role in transport. Okay, getting the idea? Is it clear in the mind? Many receptor, I wrote here insulin receptor, many other receptors are there, sensory neurons receptors, getting the things or not? Now I am just moving ahead. So I am just moving ahead. NCERT wrote a data, na? That is what? Most abundant animal protein is? Abundant animal protein, what is the name? It is written in textbook, it will come in paper. What is the name? Hurry up. Collagen. Most abundant animal protein is collagen. And most abundant protein or enzyme? Abundant protein or enzyme. What is the name? Rubisco. Rubisco is present in plant and have a role in which one? Photosynthesis. So if they are asking most abundant protein, what the answer will you give? Rubisco. If they are asking most abundant animal protein, you have to write which one? Collagen. Okay, ji. Now we are going to deal about the structure of protein. What we are going to deal? A structure of protein. So if you talk about protein, it have different level of a structure, primary structure. When you read NCERT told na, for a inorganic chemistry people, who is important? Molecular formula, MgCl2, CaCO3. For organic people, 2D structure, benzene ring, getting the things or not, two-dimensional model. For physics people, 3D approach. And if you talk about in biology, we even talk about the things not only in 3D, we also talk in 4D model. Getting the idea? Is it clear in the mind? So if you talk about protein structure, it have primary structure, it have secondary structure, it have tertiary structure, and it have quaternary structure. So tertiary structure is what? 3D. Quaternary structure is what? 4D. Got the idea? So primary structure. If you talk about primary structure, what is the reason behind primary structure? It is due to sequence of amino acid. In primary structure, two amino acid join together by peptide bond. So every protein have their different primary structure. And who decide the primary structure? Sequence of amino acid. You just try to understand with an example. I am asking a small question. How many letters are there in English alphabet? Tell me. Pin the answer. 26 letters. Very good. Now I am asking, suppose that you have gone through 3 novels. A novel, B novel, C novel. All are English novel. All are how many novel? Which novel? English novel. A, B and C. So in all these English novel, how many types of alphabet involved? How many types of alphabet involved? 26. Novel is 3 type, na? You have written 3 fiction novel. I have written the name of novel, A, B and C. All 3 novel have same story or different story? Different story. But in all novel, alphabet involved is how many? 26. Then why the different story is there? Who is behind the different story of the novel? Alphabet participated is 26 only. Now why story is different? Arrangement of alphabet is different. Suppose that different word arrangement are different, na? You can say, na? And that is the reason storyline is different. Suppose that one novel start with once upon a time. Another novel start with there was a king. So one ONCEUPONATIME arrangement is there, and there THERE arrangement is different or not different? Clear, na? So likewise, different arrangement of alphabet making different novel. In same way, different arrangement of amino acid in a protein forming different protein. Got the idea? You think like novel is equal to protein and amino acid is equal to alphabet. Got the idea? Understand the idea? So how 2 different protein differ from each other? Clear? Came in mind or not? How 2 protein are differ from each other? Due to difference in the arrangement of amino acid. Trypsin and insulin both are protein. Both are made up of amino acid. One is digestive enzyme, another is hormone. Getting the idea? How this trypsin, both are made up of amino acid. How trypsin and hormone is insulin differing? Arrangement of amino acid. So these different arrangement of amino acid is responsible for different structure and different functions of the protein. Did you got the idea, understand the idea or not? Tell me, children. Clarity came? Understood? Yeah. So now I am coming once on the primary structure of the protein. So look here about the primary structure. I am drawing the structure of the protein. Look here. N terminal, C terminal, and these are amino acid. So in primary structure, what you are finding? It have a thread-like structure. It have a thread-like structure, long thread-like structure. You just take a woolen ball, sorry, woolen thread. Wool, you have seen the sweater making wool. You take a thread of wool. Straight thread of wool is a primary structure. So here it is N terminal, here it is C terminal, and these are the amino acid, and these are which bond? Peptide bond between two amino acid. Here another peptide bond. Peptide bond. So here how many amino acid there? 1, 2, 3, 4. How many peptide bond is there? 1, 2, 3. So 4 amino acid, 3 peptide bond, 100 amino acid, 99 peptide bond. 2 amino acid joined together by 1 peptide bond, and it is called which one? Dipeptide. Clear in the mind or not? It is called dipeptide. How they are joined with peptide bond? Have a look. So I am just writing R, C, COOH, H, NH2. Here I am writing another amino acid, C, R, COOH, H, and who is there? NH2. Now what happened? Who will release? A water will remove. Dehydration occur. H2O. And what the bond will form here? It is C, R, C, O, NH, C, R, C, O, O, H, 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 NH2, and who is releasing? One water molecule is releasing. And who is the bond formed here? It is peptide bond. It is peptide bond. So two amino acids joined together by which bond? Peptide bond. Here free end is which one? Amine group. So it is called N terminal. Here free end is which one? Carboxylic group. So right side is called C terminal. So while left hand side we are saying N terminal, right hand side we are saying C terminal. You got the idea? So in a primary structure, many amino acids join together with the help of which bond? Peptide bond. An arrangement of amino acid determined by DNA. Who decides the arrangement of amino acid? It is DNA. So 2 peptide, dipeptide. 2 amino acid joined by, it called dipeptide. 3 amino acid, tripeptide. 4 tetrapeptide. 5 pentapeptide. 6 hexapeptide. More than 10 polypeptide. More than 10, we are using a term what? Polypeptide. Getting the idea, understanding the idea. Is it clear in the mind? Right? Okay. So what is polypeptide? When many amino acid, more than 10 amino acids, suppose that when 100 of amino acid join together by peptide bond, the structure formed is called polypeptide. And in a protein, number of polypeptide is either 1 or more than 1. So a protein is made up of either 1 polypeptide or more than 1 polypeptide. Clear in the mind? Is it all right? So primary structure over, done. Now we are going to talk about secondary structure of protein. It will take another 3-4 minutes, it will over, don't worry. Secondary structure. Secondary structure is of 3 type. 3 type of secondary structure is there. Alpha helix, beta plated, and third one is collagen helix. And these collagen helix is also called triple helix. Got the idea? So if we talk about alpha helix, it is what? N terminal. You just look, it is which terminal? N terminal. and here it is helically present C terminal. In primary structure it was like this. Now it become like this. It is which structure? Secondary structure. Helically coiled structure. It is called alpha helix. Now beta, any kind of secondary structure is due to which bond? Hydrogen bond. Hydrogen bond. Got the idea? So these are the folds create, coil create due to attraction between two hydrogen amino acid by their hydrogen bond. They want to come close together and they are making a fold. Now beta plated or beta sheeted just look like here. Between these two polypeptide or two chain, which bond is present? H bond is present. And it is going to form a sheet-like structure. So it is beta plated structure. H bond is still there. And collagen helix, triple helix, it is a very beautiful model given by which one? GNR. GN Ramachandran. In NCERT it is given so triple helix model of the collagen is given by which one? GNR. This triple helix is also a which kind of a structure? Secondary structure. And this structure is decoded by GN Ramachandran, a scientist from India. Getting the things or not? And the work like to got a Nobel Prize. That level of work is there. But unfortunately he not got it. It was a misfortune. Got the idea or not? So collagen helix or triple helix model, all alpha helix, beta plated and collagen helix is a which kind of a structure? Secondary structure, and all are due to which bond? Attraction between two amino acid due to hydrogen bond. Now we are going to talk about tertiary structure of protein. Tertiary structure of protein. You have seen the wall of the woolen ball. It look like this. N-terminal C, protein, long chain protein is going for a high degree of enfolding. It become like this. This is which structure? Tertiary structure. It is globular structure. Most of the enzyme are in which structure? Tertiary structure. NCERT wrote na, protein perform most of biological activity in tertiary structure. Means tertiary structure required to perform the biological activity of the protein. This is an enzyme and this is the active site. Most of the enzyme have which structure? Tertiary structure. Many fold. A thread is there. A long thread is getting folded like this and making a ball. Wool ball is a beautiful example of which one? Tertiary structure. Getting the idea, understanding the idea. And bonds present in tertiary structure is, you keep in your mind, in this tertiary structure, bond present is, first I am writing ionic bond, second I am writing disulfide bond between two sulfur containing amino acid, third I am writing hydrophobic attraction. So these all are present in which one? Tertiary structure. And every bond is a reason behind a fold. Everywhere ionic bond is forming, fold will come. Everywhere hydrophobic attraction is there, fold will come. Everywhere disulfide is there, fold will come. Fold, 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 and form a globular structure. And it is tertiary structure of the protein. Now I am telling you about the quaternary structure of protein. Quaternary structure of protein. If you talk about the quaternary structure of the protein, more than two polypeptide. This is a one polypeptide, one tertiary structure. This is another polypeptide, another tertiary structure. Join together. Getting the things by which bond? Either by H bond or by van der Waals attraction. When two tertiary structure or more than two tertiary structure join together, either by hydrogen bond or by van der Waals attraction. It is called which structure? Quaternary structure. And very good example is given in your NCERT is what? Hemoglobin. Hemoglobin. In hemoglobin, what is there? 2 alpha and 2 beta polypeptide is there. So how many polypeptide is there? 4 polypeptide is there. 2 alpha and 2 beta polypeptide are there. They join together. Understanding the idea and forming a 2 wool ball or more than 2 wool ball join, forming a which one? Quaternary structure. So 2 polypeptide and more than 2 polypeptide may join by hydrogen bond, may join by van der Waals interaction, may join by disulfide bond. So these are the possible bonds present in the which one? Quaternary structure. And these are about the structure of protein. Why bond is important? Bond is important for folding of the protein chain. As many bonds came, as many folding. I am giving you a 2 kilometer what? Thread. And I am telling and you are folding it. So as much fold, as much different bond. Bending is due to which one? Bonds. And they are going to form a globule structure. Got the idea? Understand the idea? Clear in the mind or not? So this is about the which one? Structure of the protein. Done? Now I am sending you the QR code. Have a look. So, G. Kartikeya, yeah, are you understanding what I told in this class today? Are you getting the idea? Okay, that's great. So this is your QR code. This week's syllabus, just give me a moment. So listen it. Biomolecules start from lipid, after lipid, primary and secondary metabolites, biomacromolecule, protein, structure of protein, polysaccharide, nucleic acid, DNA, RNA, nature of bonding, bond linking, monomers in a polymer, dynamic state. Means in enzyme, I am telling, start from lipid, you keep in your mind, start from lipid. In enzyme, it is coming up to factor affecting enzyme action. And 15 question in section B will come from anatomy of flowering plant. Anatomy of flowering plant. Got the idea? So 15 question from anatomy of flowering plant, complete chapter. And in this chapter, start with lipid and up to factor affecting enzyme action. That is the 35 questions will come from there. Got the idea? It's all right. So anything is doubtful? Any doubt is there in your mind? Anything you want, you not understand and I have to take care? No. Thank you very much. Bye-bye. Have a good day to all of you.